Okay, it's my pleasure to introduce our first presenter, Jon Stonehouse, Chief Executive Officer of BioCryst, to give us a brief introduction, and then we will have a Q&A session.
Good morning. Thank you, Gina, for the introduction and inviting us to your healthcare conference. With me today is our Chief Commercial Officer, Lynn Powell, and our Head of Investor Relations, John Bluth, and they'll be joining me in the Q&A session. So as Gina said, I'll be giving just a quick overview of the company and our programs, but we'll spend the bulk of the time going through question and answer with Gina and hopefully questions from the audience. It's a really exciting time to be a part of BioCryst, and there's a lot of activity going on. 2019 is a big year for us with some important milestones, and I look forward to sharing the progress we're making and our plans for the year. I'll be making some forward-looking statements. Those statements have risks. Those risk factors can be found on the website listed on slide 2. So what makes BioCryst different from a number of different other companies is we are working on bringing patients with rare diseases an oral drug. This is not some minor convenience improvement. This is life-changing for patients with rare disease, because every time they inject themselves with a needle, whether it's intravenously or subcutaneously, it's a reminder that they're sick. And the idea that they can take a capsule or a pill to alter their disease is mind-blowing to them. It's something they never thought was possible, and they're extremely excited about that potential. And as a result of all the good work that our discovery folks have done in Birmingham, Alabama, and the progress our clinical team in North Carolina has made, you can see on slide 4 the progress we've made with our pipeline. We have BCX7353, which is our lead program, an oral drug for both the prevention and treatment of HAE attacks, and I'll cover both programs. We recently, I think it was last week, announced the target for BCX9930. It's an oral factor D inhibitor for complement-mediated diseases, and I'll give a little bit of background on that. We also have a program, BCX9250. It's an ALK2 inhibitor, and we'll be moving that into phase 1 in the second half of this year, and we're going after FOP with that molecule. And then we have the antiviral programs, not core to the strategy, but clearly helpful in providing additional capital to the company. So a very full pipeline. So let me start with the prophylactic program with BCX7353 for hereditary angioedema. So I said that these oral drugs are not some incremental convenience improvement. They're life-changing. And Lynn's team has conducted multiple thorough and comprehensive market research, asking many, many questions of both patients and physicians. And the one before the one I'm about to show you was done about a year ago, before the entry of 2 new therapies, Haegarda and Takhzyro, that are more effective HAE treatments, but still injectable. And so we thought it was very important to find out how patients look at an oral prophylactic drug now that these other improved injectables are on the market. And so what you see on slide 6 is results from a patient survey that we did in November of last year, and we showed them at the beginning of about a 12-minute series of questions that they needed to answer. We showed them a profile that was similar to the profile of the results of our APEX-1 study, and then asked them a series of questions. Two of the questions that we asked is, an oral preventive HAE medication would fit my life better than an injectable HAE medication. Do you agree or disagree? 97% say they agree. And then the second question, which I find even more important, I like my current preventive HAE medication, but if an oral preventative HAE medication became available, I would switch to that new medication. And you see the medications on the bar chart on the left, and you can see that some of the patients are on the new drugs, and 89% of these patients surveyed said that they agree with that statement, and 10 of the 14 that are on Takhzyro, the subcutaneous antibody, said that they agree with this statement. So what it tells you is that patients really want an oral. And what's important in order to get it prescribed is that physicians support that. And what you see on slide 7 is the same market research, only done with 100 treaters of HAE patients in the United States, again done in November. And in all 3 of the sets of questions, you can see extremely strong agreement. In particular, the last one I think is important. When a patient requests a specific medication, I prescribe it if it's clinically appropriate. So we saw in the previous slide, patients want it, and we see in this slide that physicians will write it if they want it. So we're very excited to see that the consistency of result of patients wanting an oral drug, whether it was before new medications came out or after. So we have a proof of concept study that we're very proud of. It's called APEX-1. It was our phase 2 proof of concept study, a dose ranging study, and it was published in the New England Journal of Medicine, and you can see that reference on slide 8. I'm not going to go into the details of that. We can answer any questions you might have on that study, but it's pretty comprehensive, and it's not common to have a phase 2 study published in the New England Journal, so we think the quality of the conduct and the result are very impressive. So we're in a phase 3 study. We're on the home stretch. This is a study we refer to as APEX-2. It's on the design is on slide 9. Basically, the differences between APEX-1 and APEX-2 is it's bigger, so 32 patients per arm. We actually over-enrolled, and we announced back, I think, in November that we had hit 121. We were targeting 96, so that just shows the enthusiasm for getting into this trial. And the other big difference is that instead of a 4-week study, it's a 24-week study. So as I said, we're on the home stretch, and we expect to report out the results in the second quarter for the 24-week analysis of safety and efficacy. The body of experience is increasing. This is a slide 10 that we had shown earlier in the year. The numbers have clearly gone up since January, but the point of this slide is that with each month that passes, we're building more and more experience of patients being on our drug for a longer and longer period of time. We now have patients on drug for over a year, and what's important is that when we go to file, that we have 100 patients on each dose for 1 year, and we get that through both the APEX-2 study and then open label safety study we refer to as APEX-S, which are both running and will continue to run until and keep patients on drug until we file and get approval. So that's the prophylactic program. Now let me switch gears to the acute treatment program. Same molecule, different formulation, will ultimately be a different brand, different dose. What matters in the treatment of an acute attack versus preventing attacks is that you get rapid onset and you have a long duration. 2 very important points. Anybody that tells you that one of those is more important than the other it doesn't know the data. It's extremely important that you get rapid onset and long duration. And what you see on slide 12 is both the PK from our healthy volunteer study on the left-hand panel, and then with the 750 milligram dose, the PK in HAE patients in the middle panel. And what you see with the 750 milligram dose is that we get to concentrations of 16 times the EC50 in 30 minutes, and you stay at that level for the full 24-hour period. 16 times the EC50 is the upper limit of normal for healthy people in terms of their kallikrein inhibition. So that's plenty of kallikrein inhibition to be able to alter an attack. And what we see, when you go to the next slide, is that we got a very robust result, a very nice dose response, whether you look at the visual analog score change from baseline, and you see that the 750 milligram had the biggest difference, or you look at rescue use difference compared to placebo, you see a very nice dose response, 750 milligrams being the best. We've shown in the past a Kaplan-Meier curve that shows that that duration is sustained for a full 24 hours. And so we feel that this is an extremely successful study, indicates a robust result for the 750 milligram dose, and we'll be moving that into a phase 3 pivotal study after we speak with the regulators in the spring and start that study in the summer. So I mentioned that we have a new program that we identified or unveiled the target, which is oral factor D for complement-mediated diseases. We're extremely excited about this program. It's early, but we think it has great potential. See if I can move this slide. Let me jump ahead to the market slide. So one of the reasons we're really excited about this program is that the potential is huge. This term has been used in the past. It's a pipeline and a molecule. So with complement-mediated disorders, right now there's only one drug on the market. It's IV infused, and it's to treat 3 different diseases, and the annual sales in 2018 were $3.6 billion. 55% of that outside of the U.S., so it's not just a U.S. market. But the potential is even greater. With each year that passes, more and more complement-mediated diseases are being identified, and we believe in addition to the 3 that this drug is already approved for, there could be an additional 7. So the market potential in each of them, with the potential of being $1 billion, so the market potential here is huge, and we're very excited about this. So why is factor D important? It's important for a few reasons. First off, it's upstream in the alternative pathway. So what that does is it allows you to capture complement-mediated diseases that occur, that have defects earlier in the complement cascade. That's important. The second factor is that it plays an important role in the amplification, and that spills over into the classical and lectin pathway. So it has the potential to also affect diseases outside of the alternative pathway. So this is not an easy target. It's a serine protease, and there are over 300 in the human genome. So you can make a potent one, but you also have to make it specific to factor D. But if you're successful in making a potent and specific inhibitor, you have the potential to treat a lot of different complement-mediated diseases. So we put out some initial data around potency and selectivity that you see on slide 17. I won't go through that in detail, but there are a number of different assays. That's one of the other beauties of complement-mediated diseases, is that there are a lot of commercially available assays that you can use to get a real sense of the effect on the complement pathway. So potent and specific to factor D. And then this next slide, which is slide 18 for those of you on the webcast, is also a very important one. This is non-human primates orally dosed with 9930, and then their blood sample taken and put into a hemolysis assay. And the reason, and what you see in this slide is you see, after dosing, dramatic impact on the complement system and on hemolysis. And the reason that's important is the mechanism for the complement pathway in non-human primates is very similar to humans. So this, we hope, is a very indicative test and result of what we will hopefully see in humans as well. So very excited with these results. You can see from the PK that we've got in our non-clinical toxicology program that we get nice linear PK, and I think another really important point to this program is that the safety margin is enormous. And what that allows us to be able to do is have dosing flexibility. And I think in this disease where the turnover of factor D is extremely high, you need adequate drug levels. You need a potent inhibitor, but you also need adequate drug levels to mop up all of the factor D circulating. From a financial perspective, we're in a very solid position. We did an amendment to our existing debt that we have with MidCap early this year and changed our loan from a $30 million loan to a $100 million loan with an immediate additional $20 million onto the balance sheet of the 70 that was added, and then the flexibility to draw down more based on certain successful events in our program. So it just allows us much, much more flexibility going forward, and we're excited. It's plenty of cash to get to some very important milestones. So let me end with what I think is a pretty action-packed 2019 in particular, but 2020 as well. As I said before, the readout of the APEX-2 results, our pivotal study for prophylaxis with 7353 in the second quarter, and with success in that study, we will drive forward to file by the end of the year, which is a very important NDA filing for BCX7353 in the U.S. and then shortly thereafter in 2020 in Europe. We'll be advancing 9930 and 9250 into phase 1 this year. We expect the results from that phase 1 by the end of the year, and that's important because there's some really interesting biomarkers that even while we're looking at doses in healthy volunteers, we can get a sense of the effect on the complement system. And then we'll be moving into the phase 3 pivotal study for acute treatment with 7353 and HAE sometime in the summer. So that's the summary. I guess we'll now, Gina, move to your questions.
Thank you very much, John, and that's very helpful. So maybe I will start, with the specific timing of APEX-2 top-line data. I think that's being the most asked question from investors. So could you remind us when the enrollment was complete and how much data should we expect in the top line?
So just to be clear, we announced at an earnings call that we had completed enrollment. We never really gave the exact date. So we can't give any more granularity than second quarter in terms of when the results will be coming. In terms of what we'll show, I'm confident that investors will have some good sense of both efficacy and safety. Will we show as much as we have in the past with 7353? Maybe not. One of the things we learned with the New England Journal is holding back some information for a big publication or big presentation is important. So we may hold some back, but investors will have a very good idea of the safety and efficacy from whatever we report out at that point in time.
Okay, so will we get the actual attack rates and the differences?
Yes.
Okay.
Yeah, that's the primary endpoint. So that will be part of whatever announcement we make in the second quarter.
Okay, great. And also, based on your feedback from doctors and the regulators, what level of attack reduction do you think will be both clinically meaningful and commercially competitive?
Yeah, we've done, as John indicated earlier, a number of different pieces of market research, some before the new products have come out, some after. And we have looked at what patients and physicians prefer. We've looked at our phase 2 profile, and then we've varied a number of factors, including efficacy. And what was quite remarkable in all of our studies is that as efficacy goes up or down, share remains pretty constant.
Okay. So, there is no, if you give us a little bit color, are we talking about 50% reduction rate or 70% reduction rate?
Certainly, you're going down. We've tested as low as 50, 55%, and obviously we've gone higher as well. And yes, so obviously there's a little uptick at higher and a downtick with lower, but really relatively constant.
Yeah, I think what shocked us is that efficacy wasn't as big a driver of preference. We thought it would be a much bigger driver. And what that tells us is that patients really want an oral and that they're willing to sacrifice some efficacy for the convenience of feeling like you've got a normal life. So we believe that the trial usage of this drug will be extremely high. And so then it's our job to make sure that patients stay on the drug, that they have a good experience with the drug, and that they stay on the drug. And we're confident that that will be possible. So.
Okay. And then I think during the phase 2 trial, there is some confusion between GI tox and abdominal attacks, right, for the patients. So can you remind us the steps you have taken in a phase 3 to address this?
Sure. So just to give folks who may be less familiar a little bit of background, abdominal attacks and HAE are very difficult to kind of peel apart in terms of, is it a real attack, or do I have a GI bug, or did I eat something funny? Because the early symptoms are similar to an upset stomach. And what patients are trained to do is take their treatment early because they don't want to let it go, because if you let it go too long, the attack can become very severe, and you can end up with pain that puts you in the hospital. And so sorting out, is it a GI attack or not, is a tricky thing regardless of the study or not. So in our study, there was some confusion around adverse events, GI adverse events, and the symptoms of a GI attack. What we're doing in the phase 3, to get to your question, is a few things. The first one is we're choosing doses that we didn't see this kind of GI confusion in the phase 2 study, at least with the lower dose. And we'll see what happens with the slightly higher dose. That's one. The second one is we're going to a investigator-confirmed attack versus an adjudication panel. In the phase 2 study, there was a panel of 3 experts. All the diaries of symptoms were sent to these experts. They reviewed them in batches and either confirmed or said, no, that wasn't an attack. In most cases, when a patient gave themselves rescue therapy, an adjudicator didn't question whether or not it was an attack. With an investigator-confirmed attack, this is their doctor. So within 2 business days after they've entered into their electronic diary that they've had an attack, the investigator is to call the patient, and there's a series of questions that they walk them through. And the bottom line is they come to a conclusion on, is this an attack or not? I'll give you an example. If the patient has GI symptoms, they think it might be an abdominal attack. They took their rescue medication, but 2 days later, they're still having the symptoms. They can still go to work or school. They're taking their study medication. They're not in pain and need to get to the hospital. That's probably not an attack, and an investigator would sort that out. So we think that's an improvement. And then the last change that we've made is in having the patient take it with their biggest meal of the day. There's no food effect with our drug, and so we think instead of telling people to take it in the morning, which is probably not the biggest meal of the day, perhaps food will have some effect on the GI adverse events.
Mm-hmm. So maybe one last question for the 7353. So for ND filing, long-term safety data also will be needed in addition to APEX-2 data. Can you remind us the progress of APEX-X trial?
Yeah, so APEX-S was started around the same time as APEX-2, making very good progress in terms of enrollment there. And so combining the patients from APEX-2 and APEX-S, we expect that we'll have 100 patients per dose and with over 48 weeks, and that's what's required by the agency to file. So we're on track for that by the end of the year.
Okay, great. And then lastly, we have a few minutes or seconds for the factor D inhibitors, your new programs, and it sounds very exciting. So in the non-human primates data, I think that the 9930 achieved over 99.9% suppression of hemolysis using rapid RBC assay. So how predictive this model when translated into human?
Yeah, very predictive. So this is commercially available. It's not something invented by BioCryst, and widely accepted as a real good indicator of suppression of the complement pathway. And what's great is the mechanism of the complement pathway in non-human primates is very similar to humans, and so showing that fantastic suppression and dosing orally with primates is a good indicator for us that we could see something similar in humans.
So another question for the initial data, phase 1 data in 4Q this year. What will be your goal regarding biomarker data?
Yeah, that's, again, the beauty of complement-mediated diseases. Even in healthy volunteers, you can do the same thing with patients that were dosed orally with our drug and put it into the same red blood cell hemolysis assay. So there's a number of commercially available assays, and we'll get a really good sense of our drug's effect on the complement system, and actually might even be able to help us in choosing doses to go into patient studies.
Okay, lastly, how do you see your molecule differentiate from, say, Achillion's 2-generation molecules?
Yeah, I think both of us are too early in the program to really say, how we're different. I think the goal of our program is to have a drug where we can get to levels, that's potent and selective, and that we can get to drug levels that are high enough to be able to catch factor D in that high turnover rate. And you need a large safety window to be able to have the flexibility to dose. And so we feel we have that with 9930, and we're really excited about the profile.
Thank you very much.
Great.
Thank
Thank
you.
you.